at all for inviting me.
Okay.
For inviting me to be here. Thank you, Valentin and Anatoly and Olesia, for the opportunity to show you some of the results that our group is producing now in the field of the nanoparticles. So I have chosen today to present you something related to the synthesis and the characterization of gold and silver nanostructures and their application basically in antimicrobial studies and in some catalysis devoted to degradation of colorants or organic matter. So, but before to go ahead, let me to show you something about the institutions that are involved in this work and explain you which institutions are. So the Bioscope Research is the name of our research group, and we have a society, a scientific society belong to us that Anatoly Olesia can know because it's the society that organized the international conference, is the Proteoma Scientific Society. And then we belong to a big organization of laboratories in Portugal that is called Requinte. This is a laboratory devoted to green chemistry and sustainable chemistry that cover in different universities from the north, from Aveiro, Porto, Coimbra, Trás-os-Montes, also Lisbon, that we are the node of Lisbon, and in the south we have Évora. So it's a network of a network of laboratories. In this moment, we are around 1,200 researchers in all of these institutions. And then below to the Nova University of Lisbon, and we are from the Nova University of Lisbon Faculty of Science and Technology. And this picture that I show you here is the main entrance of the university. Here we have at the end the building of the mathematical department, and here in the front the building of the mechanical engineer department. So what happens with us at the laboratory? So we are a multidisciplinary group. We are not all of our teams are working in nanomaterials. So we have biochemists, chemists, material scientists, physics, we have biologists, we have chemical engineers, all of them working in different subjects of the field. So we create the nanomaterials in the laboratory, starting by bulky materials, in our case are metals or salts, and then we produce materials that will be sent to the other lines in the group in order to test it. For example, as analytical tools for proteomics or proteobiomolecules, or for example, in catalysis or in uses as tools for example, connecting organic molecules in dyes or in pigments or in sensors. In general, we collaborate with people around the world. Here in this slide, I show you in blue the name of the members of my group that are related to this research, but also the international collaborations that are devoted, some of them in some universities of Spain, in Madrid or in Galicia, other universities in Europe, like the Bulgarian Academy of Science. We collaborate a lot with them. Also with the Bucharest Polytechnic University of the University of Antwerp in Belgium. And out of Europe, also we collaborate with the University of São Paulo and the University of Campinas in Brazil and the University of Miami in the United States. Why? Because in some of these points we need to characterize or we need to apply some of these materials or research that we don't have here in our laboratories, and then we collaborate internationally to complete the research of our papers. So just to give you an entire view, the Bioscope Research Group is working actually in 6 different research lines or different boxes, all of them connected. So the yellow one that I will show you are responsibility of my colleague, Professor José Luis Capello, and Dr. Hugo Santos and Dr. Luis Carballo, and they are working specifically with mass spectrometry with high resolution applied to environmental samples, to bacterial characterization, in food chemistry or food analysis, but especially we are working in applied proteomics to biomedical approaches like biomarker discovery, in personalized medicine, specifically focusing cancer and aging diseases. And actually, we have a very new or very recent research line in which we are working in paleontoproteomics, in which using tooth and bones from the old periods of the humanity, we can detect or we can characterize the age, the gender of the individuals, and also the illness of the individuals pass away. And today I will introduce you a little bit about the 3 other boxes that are in blue or in green. They are under my responsible. And then I'm working also with Dr. Oliveira, Dr. Duarte, and Dr. Fernandez Lodeiro, in which we develop organic molecules that we can use as sensors for detection and quantification of pollutants, for example, anions of metal ions, applying in environmental remediation and also for biomedical application. But mostly today, I will show you just the inorganic part in which we prepare metallic and soft metallic nanoparticles useful for optical, drug delivery, and antimicrobial studies. Our campus, so our laboratories are divided in 3 groups. So as you can see here, we have 2 laboratories completely devoted to the synthesis and characterization of nanomaterials. Other 2 laboratories, in fact 3, but our 2 biggest one are devoted to mass spectrometry and proteomics. And we have a middle laboratory that is devoted to antimicrobial studies with 2 of them, that following the connection between the bio part of the group and the inorganic or organic part of the research line. Our laboratories, I show you here some of the images, has pretty well conditions from the synthesis to the characterization, especially for the characterization of mass spectrometry. We have 3 equipments of high resolution mass spectrometry. We have several equipment for purification, including HPLCs, nano HPLCs, and then all the spectroscopic systems for characterization from the steady state to the excited state part of our molecules, of our nanomaterials. The campus is one of the biggest campus in Lisbon. So I show you here the picture of the Caparica campus. Is located more or less 2 kilometers from the Caparica village and 10 kilometers from the center of the city of Lisbon. The chemistry department is the biggest one that you see with the B form at the right part of this slide. This building is a multi-departmental building, so we share the chemistry department with the biology department, with conservation and restoration, chemical engineer, environmental science, and biomass production of biomass and bioproduction systems. So the campus is integrated by almost between 80,000 and 10,000 students, and we are around 550 professors, and taking into account the researchers, we will be around 1,500 individuals. The campus is very close to the sea. As you can see in the top of these images, this is the Atlantic Ocean. And the city you can see in the left part is the Caparica city, in which we organize the conference or the seminars of the courses devoted to our research line. So one of the most impressive things we have here is the light. Lisbon is a city that they call it the city of the lights. It's not Paris. I'm sorry for the French people. But in summer, one of the most nicest things we have are the sunsets. And this picture was taken from the balcony of the hotel we organized the conference in front of the ocean, with one of the typical sunsets we can see in June, July, and in August. So just moving now to the scientific part of the conference, to starting to use. So always we start with a metal. And regularly we work with metal salts that will be soluble, especially in water. Then we introduce organic molecules. These organic molecules many times are used as a fluorophore that could be excited or could be antenna effect, taking some photophysical properties from the metal. Others are just using to stabilize the molecules. Others are used as a reductant. So some cases we use a reductant. In other cases, the organic molecule will be also the reductant in order to reduce the metal ion and produce the nanoparticle. So we are working now especially in anisotropic nanoparticles. So for those of you that are far from this topic, let me to introduce you what is an isotropic nanoparticle. So an isotropic material is something that is not spherical. And this is very useful for us because you can move the photophysical properties very far from the visible. So the characteristics of the nanoparticles are directly dependent to the form of the shape of the nanoparticles. We can use or we can explore the optical properties, the electronic properties. We can apply these materials in catalytic properties, especially those that are involved with palladium and platinum. We can also working in the field of magnetic nanoparticles, applying with iron when we create the gold and the silver nanoparticle with an iron core. And also we can study, especially for the biomedical applications, the precise control for drug delivery or for interaction with a protein or for interaction with a peptide. In our group, we are actually working in 5 different applications. Each research line is devoted to one of them. So biological imaging and diagnostics, we are working with these nanoparticles, especially those that have the plasmon resonance bands in the near infrared region, just because they are very useful to excite it without damage any kind of cells or tissues, and they are very useful for vivo applications or for drug delivery or targeting therapies. We use them mainly as a sensors because we can highlight the properties of the organic molecules when it's connected to the metal core. We can use also for catalysis in material science and especially in drug delivery. In this moment, we are able to create different morphologies. And in this picture, I show you some of the materials we are able to prepare from nanorods, nanostars, nanotriangles, nanosquares, working nanoparticles or polyworking nanoparticles. And also we can play with the composition of these metals. We can working with a pure metal with gold or silver or palladium or platinum, but also we can prepare it alloyed nanoparticles, intermetallic or subcluster nanoparticles, and then playing with the most useful for us, the multi-core shell nanoparticles. These shell nanomaterials can be also connected in some cases with silica, mesoporous silica or crystalline silica, in order sometimes uses as a layer to release or to reduce the release of the metal ions from inside to outside of our nanomaterials. For example, I select this paper to show you just because it was a surprise for us. We were using these organic molecules, is the phenyl ditelluride molecule, a very simple one, in order to create silver nanoparticles. We has a collaboration with University of São Paulo in Brazil, in which there are a very strong group in the developing of different chalcogenide materials, especially devoted to selenium and tellurium. And with this, and when we mixture with a silver solution, for example, with a nitrate of silver, we create a material that don't emit light. So this material was characterized by high resolution transmission microscopy and other techniques. And you can see here, instead, the single nanoparticles as what at the beginning supposed to create was formed a kind of a network of fibers. These fibers has dark fields and white fields along them, so we can also observe it where the metal are located and where the organic part are located. The solution is a red solution, but it's completely blank, so don't emit light. And for us, this was a problem because if you like to use this material for imaging, you cannot see absolutely nothing. So with them, we decided to modulate the properties of these materials in order to check what happened with the silver when the silver will be also reduced. So, and here we have the results. When we reduce the silver in the complex of the materials to silver nanoparticles, the solution becomes more red and then highly emissive. In the solid state, the fibers are strong emissive, and in the solution, the emission in concentration is yellow, and the emission in the more diluted solution are completely red. So these particles that are formed are in the level of microparticles with an intensities around 230 nanometers around. However, it's very interesting because we can now, one modulated the system, we can recognize by these single dots white present in the center of this slide that are the silver nanoparticles. And in this case, we observe that are not completely spherical, but are not completely silver nanoparticles. We observed that these emissive materials are formed by a silver tellurium quantum dots, and these silver tellurium quantum dots are very sensible, for example, to the temperature. We can modulate the temperature up to 100 degrees and is increasing the emission. This increased emission is because the aggregation between the material and then is completely quenched after achieving around 150 degrees. The material is almost reversible. We can achieve the middle of the intensity that we started, and then the emission remains in the same position. how we can modulate this, how we can change it with amines, for example. If we use oleilamine, the oleilamine produces the red color solution and the emission is the highest intensity. If we use hexadethylamine, so we can increase the length of the chains, we produce a very low emissive material. But for example, if we use decanothiol, in which we introduce a sulfur atom, we quench it completely the emission, and is the red line, and is the blue solution that is completely plain. This was very interesting because we observed also, because of the presence of the silver, some antimicrobial effects that killed some of the bacteria. But the problem we obtained in this system is the solubility. The fibers, after being solid, is very difficult to dissolve and very difficult to use for bio applications. So in that case, we decided to remove the tellurium reductant ions, and then to use other kind of molecules. And this paper I show you here was published very recently, in which we use adenosine monophosphate, AMP, also works with ADP and ATP, and is a biological molecule, very compatible, the final material, that in the presence of EDTA, we can play with the pH, and we can, for example, produce anisotropic materials in a way of control the dimension of the materials. In this case, we produce nanoplates. That this is in these triangle nanoparticles can be also growth in the thick size and in the long size. So as you can see here, working with the pH between a pH 8 and pH 11, the absorption band of the nanoparticle is modulated between 420 and 1,200 nanometers. So this allows you to obtain nanoparticles that are very stable in the near infrared region. But also, if you work with the multi-growth methodology, in which we increase the concentration of the adenosine monophosphate, we can move the plasmon resonance band close to 2000 nanometers. And actually, we are using these materials, for example, for antibiotic effects, for the killing bacteria. They are working pretty well. These are samples, these are results that I cannot show you here because are still in progress, and the paper will be submitted before December. But this is very good because we can irradiate our nanoparticles in a regions with nothing absorb the light. So we can prevent the killing the bacteria, or is in the, for example, using the protocol for cells, we don't kill any part of the materials. So they are very monodispersed. I can show you here in these slides in these pictures of transmission electron microscopy, and just playing, for example, with the pH, we can control in this case the plasmonic band and then the thick of the nanoparticles. They are pretty well organized, as you can see in the panel F, in which we can see that each plate are connected over the other, and then changing also for these supramolecular interactions the photophysical aspect of our nanoparticles. Sorry. So maintaining the same synthetic approach using the adenosine monophosphate, we can also create nanourkins. These nanourkins has been protected with an external silica coating shell in order to reduce the time of release of the silver ions. You know that one of the antimicrobial effects of the silver nanoparticles are because of the silver ions released to the medium. However, many of these nanoparticles are are easily hydrolyzed, are very unstable in a water medium, so you need to control the release of the silver ions. In that case, just putting a silica layer around the nanoparticle, and you play with the length and the thickness of the silica layer, you can reduce the time, and then the nanoparticles live a long time for the biological or for the macrobiological applications. In this case, I show you two pictures just to demonstrate the highly monodispersity of the nanoparticles, and also the highly monodispersity of the layer we can create around the nanoparticle. These layers are between 15 to 22 nanometers, depending on the methodology we use. This system was, for example, tested with water that are contaminated with a bacteria that is the Rucivibrus gelatinosus. This bacteria is a biomarker control for the quality of the water. And then you can see in the control example is nothing, so we don't see absolutely nothing. But then just we added the nanoparticles, adding the nanoparticles from 2 to 10 micrograms milliliter, we are starting to see the growth of an halo around the nanoparticles that inhibit and kill the Rhesus gelatinosus bacteria. So we are also exploring this in contaminated waters, and we are working in this field in this time. However, this is a solution. And if you go to practical applications, we need to encapsulate the nanoparticles or entrap the nanoparticles in other materials that be easily handling. And in this case, we are working with different polymers and copolymers. The results I show you here was using a polyurethane. It's a Irogran, in which we introduce the nanoparticles with the silica mesoporous in order to use these flexible materials as antimicrobial tools. For example, if you like to cover a table, or in a hospital you like to prevent the contamination in a chirurgical block, you can use these covered polymers in order to reduce or in order to eliminate completely the growing of the bacteria. So I show you here the aspect of the polymers. So the polymers are very flexible, maintain the properties of the polymer. Depending of the concentration of depending the material, the color of the polymer change. You have here the blue one from the triangles, the yellow one from the sphericals, the green one from the near infrared materials, or also if you like to obtain it in the similar way with the iron nanoparticles, we obtain the black polymer. If you see through a light, you can see there are spots in which you can concentrate the nanoparticles, and then look into the surface enhancing microscopy, you see that there are the huge number of nanoparticles concentrated in the network of the polymer, and also in these high resolutions you can see the layer of the silica, the core shell of the silica maintained inside of this polymer. The most important thing is, even after the application of these nanoparticles in a huge concentration, you can observe it like in the panel B, that the polymer is completely flexible. Then you can use this polymer as a starting material to create every material you like with this polymeric film with the property of to be antibacterial. The panel C is showing you that the polymer is porous. So have a porous, and this porous allows the release of the silver ions from inside to outside, and then they can use, for example, that could be used for microbiology applications in a hospital. The other panels are obtained with different concentration of nanoparticles. As you can see, in a time that the concentration is enough, they create a kind of nanostructures communicating the different nanoparticles between them in this kind of nanowires. So we use these materials, for example, for bacteria, just putting a piece of the polymer over the slides with the bacteria. We studied with Escherichia coli, with Enterococcus, and with enteric and with Bacillus cereus, and we observed in the 3 cases that the concentration, the minimum concentration, is pretty very well, is reduced, and the most appellative one is those that use the nanoparticles with negative charge. As you can see in this table, we can use it negative and positive or neutral nanoparticles, and the negative one connected with the silica and silver are those that reduce a lot the minimum concentration inhibitory. But then also this can observe it in the third panel, in panel C, that the Salmonella enterica and Bacillus cereus are those bacteria that are completely disappearing from this solution. So we are now applying these polymers, for example, to create smart containers for food, because prevent from the contamination of the Salmonella enterica. So moving then to another kind of nanoparticles, including changing the metal, we decided to create polystars nanoparticles, and was not possible with the silver. The silver growth with the nanoplates of the nanospheres, but not with the nanostars. So we change here to gold, and then we will see some examples in which we alloy the gold with silver, alloyed with platinum, platinum and silver, or platinum or palladium and gold. So we can see here some of the examples in which we create gold nanoparticles using iron as a reducting ions. In that case, depending on the reaction, we can modulate the anisotropic of the materials. So I show you here, if we observe it, if we use, for example, a naked solution in which we add the gold and the iron, we produce a kind of inorganic shell around the gold. This inorganic shell is formed by crystals of iron sulfate. If we use an organic stabilizer like PSS, we can obtain it, the nanoworkings, nanoparticles, and we also, if we add it at the same time citric acid, we can change in these nanoworkings as a nanoparticles. So in the same reaction, we can using non-toxic reagents, very cheap reagents, working in a very low mill temperatures, we can produce in one synthetic 3 different nanomaterials. The use of polystyrene sulfonate as a stabilizer was in this case very useful in order to create the anisotropy in the nanoworkings. The problem was later because this PCS molecule is not safety, so has some toxicity that reduce the applications that we can use it here. In fact, these nanoparticles that were produced actually are using as a SERS substrate for sensing, for optical sensing. In this case, I show you just the detection of the 4-nitrophenol molecule. This nitrophenol molecule is attached to the surface of the nanoparticles. As you can see, the fingerprint of the SERS spectra, we can go until 10-10 and 10-10 limit of detection, in which the spectra observed the typical signals for that molecule. So in this case, now in collaboration with University of Vigo, with the colloidal group, we are exploring this kind of nanomaterials for more molecules using as a active SERS substrate for detection in environmental samples. However, as I told you, the PCS was highly toxic for our applications. We decided to move this stabilizer, and the example I show you now was using polyallylamine. Polyallylamine is a molecule that is less toxic, so in some cases, depending on the pH, is biocompatible. And once again, playing with the pH, we can, using the seed methods for growth, we can create different anisotropic material. The most interesting thing is, depending on this pH, we can modulate the color of the solution. Then you can modulate the plasmonic band from the visible red region again to the infrared, going up to 2000 nanometers. So the nanoparticles were characterized by high resolution microscopy and for other techniques. And you can see here that as soon as we increase the seeds of the nanoparticles, we elongate the arms of the nanostars. Here I show you a tomography, a picture of this nanoparticle. The most interesting thing is during the synthesis, there are some holes, some defects on the nanoparticles that can be explored later to entrap some organic molecules. And we are now within collaborations with University of Angers in France to try to use these inorganic nanoparticles to entrap some organic molecules that can be used later for sensing or for imaging. So this particle has the positive property that can be functionalized via carbon diimide. So any molecule, any organic molecules you are thinking that can react via carbon diimide reaction, they can be functionalized with these nanostars. They are very soluble in water, and we can modulate the visible band, the plasmonic band, from the 580 nanometer, from the pure spherical one, to 100 to 1250 nanometers from the blue one, which we were mainly at a 5 pH. These nanoparticles also can be stabilized with adenosine monophosphate, and this paper was the results with the stabilization of the adenosine monophosphate in the surface of the nanoparticles. We started to form an inorganic complex. The inorganic complex formed by gold evolved with the coordination of the MEP, and after the coordination with the MEP, reduce the gold 3 to reduce 1 to gold 1, and then the gold 1 is reduced again to gold 0, producing the nucleation of the nanoparticle. So here I show you an images in which, in this case, we can also maintain a very stable with a very long tips of the nanoparticles, maintaining also the plasmonic band close to the infrared, in which we can achieve 900 nanometers. The spectra of the study, the full characterization, give us the possibility to study the deposition of the atoms in these tips, as you can see here by aberration correction, high resolution transmission electron microscopy. These atoms are pretty light connecting around the tips. As soon as we add seeds of the nanoparticles, we grow and we have uh, positive things in order to grow the tips of our nanoparticles. This system was used, for example, for degradation of the TMB, in which in the presence of the H2O, which can degrade and oxidize the colorant, producing the oxidase steps in which we observe the blue color, and this acting as a catalytic artificial peroxidase molecule, in which we observe one of the highest values for the Michaelis-Menten curve and for the Lineweis-Burk plot, in which we observed a very huge velocity and a very huge constants for a gold nanoparticle. However, it was not enough, and then we need to move forward with this. And we create something in collaboration with the part of our group that is working in biochemistry, in the proteomics, and to create sensors. For this case, we use these nanoparticles for the obtention of a sensor for glucose in useful and readable with a mobile phone. So with this case, we use the gold nanoparticles, in which we introduce the ascorbic acid and the molecule. We create the nanoparticles in a paper, and this paper can be injected via, for example, via inkjet printer. So we can use our solution as an ink, and then the ink solution is deposited in the paper, and then we can create a shape around the nanoparticle. As you can see here, the papers that is growing the nanoparticles inside, we obtain a plasmon of this paper around 700 nanometers, and we observe all the fibers of the cellulose present in the paper. We can grow continually cover with the nanostars. With this, we study with different glucose sensors, and in the presence of oxygens and oxidized water, we can perform an analytical detection colorimetrically of the concentration of the glucose. In order to have an application, we test it with urine, and the testing is very stable in the presence of urine without any treatment, and then we can maintain it, the colorimetric detection of the glucose, going until 70 micromolar of glucose in urine samples. In our institute, one of the most important research line from the point of view of wind chemistry is the catalysis. And from this, we study or we apply many of our materials in order to use these simple metallic or the mixture metallic nanoparticles for environmental catalysis. So in this case, we are starting with the gold nanoparticles I showed you before with the nanostars, but we decided to mix the gold with other metals that could increase this characterize. For example, if you create the nanoparticles with gold and palladium, we obtain the same shape. We can modulate it, in which the palladium is located outside of the nanoparticles, as I show you here in these images. The stem images show you in the red dots are the palladium atoms, and inside on the stars are the gold atoms. So this bimetallic nanoparticle was again tested as a catalyst and works better than the pure gold nanoparticles. However, it's not done best than the gold platinum branched nanoparticles. We can apply the same technology. We can use the same molecules to stabilize these bimetallic nanoparticles. Here in the panel B, you can see the shape of these anisotropic nanostars or nano arms with the platinum. And then once again, we can control it, the deposition of the platinum around of the external part and in the points of these nanostars. And inside, we have the gold connected into the core of the nanoparticles. Comparing the gold nanoparticles with the platinum one and the palladium, we can observe it that for the degradation of the TBM and in the presence of hydrogen water, we can observe it that we can decrease this, we can increase the velocity, so it's faster, and then we need to use less materials in order to obtain the same degradation systems, with increasing also the Michaelis constants for the same materials. Finally, we decided to reduce the cost of our materials. This is another point that we need to think. And when we work with palladium, platinum, gold, and silver, we can thought that the cost of the bulky material then to create industrially is not the best. So we changed the gold for copper. And in this case, we produce the copper one as a reducing agent to produce platinum copper oxidized catalytic nanoparticles. So here you can see the shape of the nanoparticles still remains the same. The size is around 80, 18, 9, 20 nanometers. And then we can see here in these panels that in the presence of hydrogen peroxide or in the presence of sodium peroxide, we can degrade completely. In this case, it's rhodamine blue, rhodamine B or the rhodamine blue. We can eliminate completely, in that cases, up to zero, the presence of the organic in the water. So changing by copper, the other metal, but maintaining one of the noble metals like platinum or palladium, obtained better results than in the case of the copper platinum or copper palladium. See, as I showed you, that they perform the same properties of catalytic activity depending of the pH. pH 10 is the best to perform these properties with this material, and then we can use it depending of the platinum, the highs of platinum located. We can observe it that as higher percentage of copper in the planes of platinum, we obtain better activity, and we reduce or increase the velocity. Finally, many of our colleagues ask, this is nice, but you are working in a very small volumes of solution. So how we can produce this material in scalable? So very recently, and in collaboration with University of Oviedo, we are starting to escalate the production of these gold and gold platinum or gold palladium nanoparticles. In this case, we can use it, for example, up to 1 liter of the nanoparticles. We maintain almost exactly the same plasmonic bank and the same shape of the materials, and studying the catalytic effect is exactly the same. We has added a novelty in these nanostars in which we can protect it as well, like happens previously in the silver one, with a layer of mesoporous silica or crystalline silica. And you can see here the monodispersity, and the most important is the presence of just one nanostars inside of the single nanoparticle, maintaining the plasmonic band, maintaining the extinction coefficient for the nanoparticle. So in this moment, we can create with this up to 1.5 liters of nanoparticles that are stable for more than 1 year in the bench and not lose the properties, the catalytical properties. You can see here at the end, for example, in the panel of the last, this is the degradation of the methylene blue, and you can see here as the 350 seconds, we are completely degraded, in less than 10 minutes, degraded the organic molecule. So ending again to the laboratory, we produce the nanoparticles, but not only to produce the nanoparticles, also to apply the nanoparticles. So if any of you like to collaborate or use some of these nanomaterials in your research, just drop me an email and we can speak about the potentiality of use not only in the biofields of applications in microbiology or in proteomics, but also in catalysis or in optical materials. So this is our research group. This is not all, but all they are is here. So we are in this moment 28 researchers. Here is a part of the group that are more involved in the research I present you today. And if you give me just 2 minutes more, I will introduce you the two conferences, the biggest conference we are organizing in this field for the next year. In the first is the conference of in January, the International Conference of Nanoparticles of Nanomaterials. In this moment is almost filled, but we still have room for some shotguns or from posters. And in the right side, we have the chromogenic and emissive materials. You can see here the name of Anatoly is not here in this poster because this poster comes from July and Anatolia Thet during August. So we'll be here soon, and I will send you all of you the chromogenic and emissive materials, as well as the ISNA is in the 7th edition. So we are already 14th years putting connecting the people and preparing networks of researchers. That is one of the most interesting things on this conference, because the people connect during the network sections and then can apply for international projects. Let me also to introduce you, because maybe from your interest, a journal that we launched this year. This journal devoted to translational chemistry and interface journal is a journal that have chemistry as a base, but accept papers from any kind of research in which chemistry is important. Materials, supramolecular materials, pharmaceuticals, engineering, working with energy, whatever. So as you can see here and read in the abstract of the journal, is something in which chemistry is essential to have your research or to have your results. So you are welcome to submit your papers. We have already the first issue ended in July, and now we are preparing the second issue that will appear online before December. As an editor of Dyson Pigments, that many of you knows me about that also, just to give you here some information regarding the journal. In this moment, we are one of the biggest journals devoted to dyes, pigments, and applications, and is the one that have the highest and the quick answer for accepting the manuscript. So I invite you also, if you have some results, to apply to or submit to Dyson Pigments. And finally, ending again to the sunsets in Caparica, I left you here some of my contacts and the social media, if you like, and we stand here, as I told you before, for peace. So Slava Ukraine, and thank you very much.
Героям слава. Thank you very much for your very nice lecture, very interesting about nanoparticles, material science. So, dear colleagues, we have time for questions. And please.
May I answer a question?
Yeah, sure.
Hello. Thank you very much for your very interesting report. I have some question concerning your complexes with polymer matrix filled with silver nanoparticle covered with silicon. Yeah. You say that your approach could delay the silver ion release as one of mechanism of antimicrobial activity of these materials. But how can you answer the question whether you study the kinetics of ion, this ion release, how long it could take to release ion? And the second aspect, if you cover the nanoparticle with silver carpet, it could remove any other mechanism, for example, any Ross reaction on the surface of nanoparticles. And this could be critical for antimicrobial effects.
Yes.
For example, peroxidase activity used so for other materials.
Yeah. So thank you very much. This is true. So first, the time you can control the release depends of the concentration of the particles you are using for the polymer. Okay. So in compare with the naked nanoparticles without the silica layer and without introducing the polymer, we observed that you can pass from minutes to hours. So you maintain the nanomaterial more stabilized without any hydrolysis when the nanoparticle is inside of the polymer and covered by the silica. This gives us the opportunity, for example, if you like to use the material for a long-term uses, for example, like a polymer over a table, you release the silver ions during hours or during weeks until disappear completely the nanoparticle. Because silver nanoparticle has the problem that working with them in an open atmosphere, they are very unstable. They reduce it. And then they oxidize it and oxidize to silver ions. Using this technology, we reduce this time and then give us the possibility to work in hours or in weeks. Okay? Related to the mechanisms, we don't prevent the ROS. So we have many mechanisms simultaneously. So we have, in fact, we observed with the different bacteria that the fact to put the nanoparticle inside of the polymer, we increase, in some cases, more the ROS mechanisms than the oxidizing mechanisms, the other oxidizing mechanisms. So depends of the concentration. Okay, this is not fully studied in this moment because the paper we published was the last year, and we are actually in the microbiology laboratory testing during long times the different polymers in order to detect the mechanisms and to create better materials to delay or not the release of the ions, of the silver ions. And this is a good question because you cannot control only one mechanism. You have several mechanisms for killing
Yes.
the bacteria at the same time. Yeah.
So silica prevent you the condition to transfer the ion and other reaction because
Exactly.
silica is porous. Yeah?
Exactly. If you use the silica mesoporous, you have a kind of a sponge. It's porous around the metal ion. And then the time in which the ion goes outside is reduced because must cross all the pores. And this is one that you can working with mesoporous silica, with polymorphous silica, or with crystalline silica. Depending of the silica you use, you can release faster or slower.
Thank you. May I ask one more question?
Sure.
Yes, please.
Concerning your B-metal catalytic materials, do you have experience with working with silver and superconductor nanoparticle? Such
Not now.
complex for Polaron-induced photocatalysis.
Yes,
Very
yes.
interesting approach.
Yes, not by now.
No,
Not by
thank
now.
you
It's
very
something
much.
that we like to do, and I hope to do in the next future, but not by now. Because the problem is, uh, as you change the metal, the synthetic procedure is completely different. So in fact, the results you are looking
Yeah.
here in this talk, that is 40 minutes, the results summarize almost 6 or 7 years of synthesis. Each person is devoted to one metal. So I have one devoted to silver, other to gold, other to palladium, other to platinum, and so on. Because if you change the metal, this is a simple coordination chemistry. So you change the metals, you change the orbitals, and you change the capacity of coordinate with the different organic molecules, even with the silica. And then you need to control the steps. Sometimes the reactions of control the step is easy. In other case, it takes some months in order to have the stable materials. But this is something that we like to do. So we are now thinking in move to some conductors and semiconductors connected with the silver. It's a good idea.
Thank you very much for your very interesting report and your kind words for our Ukrainian scientists working here in Ukraine. Thank you very much.
Thank you very much. Thank you very much.
Thank you, Svetlana. Vladimir, please.
Yes, thank you. Thank you, Carlos, for your presentation. I have two questions. First one, according to your experience, did you try to compare efficiency of antibacterial actions of metallic nanoparticles if they're illuminated, for example, with a light corresponding to plasmonic resonance?
Yes. Thank you very much also for this question. So in fact, we have been working during the last 6 months on this. I don't present here nothing because it's not published. One of my researchers moved to the University Autonomous of Barcelona in Spain. And then we used this silver and these triangle silver nanoparticles and these nanostars of gold and silver in order to apply the PDT, photodynamic therapy, in order to irradiate it with different lasers that go from the red to the green and uses for the different bacteria and the different cancer cells. quickly, I can tell you that the results are very promising, are very nice. And in some of the cases, our level of degradation, of killing the bacteria or killing the cell is almost 100%. So
Okay.
it's really good. Even at the beginning, we were very upset by, in some cases, by the solubility. But
Mhm.
even the nanoparticles stay stable in some cases with many EPIs or other medium that can use it for biomedical applications, and then they can be used. I hope during the next 3-4 months we can release these papers, and I could send you after.
Okay, thank you a lot. It means that you think that the main mechanism is related to a photothermal effect and
Yeah.
not, for example, to a photo-induced charges appearing in the nanoparticles. Because for photothermal effect, if I understand well, you need some quite important light powers to be applied. But if the light power is very, very small, especially for anisotropic nanoparticles, for example, we can suggest a mechanism related to appearance of the charges you see at the extremities of the particle. I don't know
Yes,
if
you are right. You are right, because in fact the majority of the work was done with lasers very, very weak, with a
Okay.
very weak lasers. And then we have exactly the same results that we can obtain it with a professional laser. So that means that it's not thermal, means that it's
Okay.
something more that is devoted. And probably in the tips of the materials,
Yeah.
we are creating a multi-charge
Yeah.
systems, and then the multi-charge systems are able to kill then
Yes.
the
Yes.
bacteria
Yes.
or the cell. Yeah.
Okay. Excellent. This is a really very nice effect, and your response is very important. Okay. The next question, also a general one. If you compare, for example, I don't know if you had an opportunity to apply your metal nanoparticles for surface enhanced Raman spectroscopy, for example, and if yes, uh, is any advantages of nanostars compared to spherical particles for SARS applications?
Yeah. As much as anisotropic is present, is better for this effect. So we compare our nanourkins that are almost spherical with a very
Mhm.
small anisotropy with, for example, the MEP synthesized nanostars, and the sensing, for example, using the SERS is higher. So
Okay.
the quantity, we reduce the amount of the analyte we can sense, but also we reduce the amount of nanoparticles used for the sensing. This is true.
Okay.
This
Okay.
is
Thank
true.
you very much. Thank you very much.
Thank you very much.
Thank you. Professor Kirichenko, please, your question.
Thank you. Thank you very much for very good presentations. My question is about anisotropic nanoparticles and thermodynamic. From thermodynamical point of view, from entropy point of view, most stable shape of nanoparticle is supposed to be spherical one. So my question is, have you checked stability of anisotropic nanoparticle over time? For example, after 2 months, 2 weeks, how stable they are?
Yes, yes. Thank you very much. So even for example, the silver nanotriangles, they are stable in solution if you prevent the oxygen, stable in solution in a flux, in a regular flux for more than 6 months. If in the case of the gold nanostars, as I told you in the last slides, we are actually studying solutions that has been prepared almost a year ago, and the nanoparticles are exactly the same. Maybe the intensity of the plasmonic is reduced between 5 to 10%, but in general, it's exactly the same. The stability in compare with the spherical nanoparticles. Of course, we have spherical nanoparticles that can be used 3-4 years after if the stabilizer has not the aggregation effect. So stabilizer prevent the aggregation. But in compare with this anisotropic that has better properties, especially for biomedical or biological applications, up to 6 months are okay. 1 year maximum we are studying now, and the results is pretty nice. So it's not changing. For example, I will give you a practical example. We use the gold nanoparticles for separation of peptides in a digested sample of blood or serum in order to then, by mass spectrometry, catch individual peptides that can be used as a biomarker, for example. If we use freshly nanoparticles prepared 2 hours ago or 1 day before, the performance, the analytical performance is exactly the same as is the nanoparticle have, for example, a month or 2 months. Exactly the same. Are more stable the gold one than the silver one. The silver one is depending on how much on the conditions. Many of them is they are not protected by silica or they are not protected by the polymer. After a couple of weeks, you are starting to see degradation. For example, the triangles are starting to disappear, the tips, and then converted in a spherical one. But this after 2-3 weeks with this synthesis. In my opinion, this is depends of the presence of the AMP. AMP is connected to the surface of the nanoparticles in such a way they could be protected the nanoparticle. This is a good in good hands, in one hand, in the other hand not. Because if we like in some cases to remove the AMP and introduce another molecule, the force is higher because the connection is very strong, protecting the nanoparticle. But you can use it. You can prepare it and use it up to 6 months perfectly.
Okay, thank you very much. It's very impressive. Thank you.
Thank you very much.
Thank you. Professor Kamarov, please.
Much, Carlos. Fantastic results and nice presentation. My question is, how did you obtain the pictures, the tomographic pictures of your gold nanoparticles? What was the method?
Yes, we collaborate with an institute of nanomaterials that is located in Braga, in Portugal, in the north. That is the Iberian International Nanomaterials Laboratory, the NL. And they have a high-resolution STEM instrumentation, and then prepare thousands of images, and then using some programs, they overlap the different images and construct the 3D dimension of these tomographic studies. As I say, you are very expensive, really very expensive. For example, the picture you saw about the gold nanoparticles, 2 samples cost around 5,000 euros. It's something that we
They
do.
look great. Yeah.
Yes, something that we do only in the cases that we have all the experimental that fits well, and then we like to see exactly how is the 3D nanoparticles that you can perform. There are a group also in Belgium, that is the group of Sarah Valls in Antwerp University. We collaborate with her also in a couple of papers. They are able also to do that using the different high-resolution microscopy, the STIMs, and then they can collecting the pictures, and like in the human body, then we can create the 3D dimension.
Great, thank you.
You're welcome. But you must know in connection with these questions. In Europe now, the commissions have different laboratories around the different countries in which there are programs. If you apply for these programs, you write a very small project. If the project is accepted, then you can have the studies for free. You don't pay nothing. And the ENL in Braga is one of them. There are another in Zaragoza, in Spain, in Cádiz, in the south of Spain, Belgium, and some of them around Europe. So you can check, this is InfraChip, the name of the project. You can check this in internet, and then you can apply for different studies for free.
Much, yeah, noticed that.
Yeah, okay, thank you. Anatoly, please.
Yeah. At first, I would like to say thank you very much for your exciting lecture, and I have one very short question because some of my questions have already answered. As I understand, you synthesize silver telluride nanoparticles, quartz shell with very wide spectral range, from visible to pretty far infrared. Do you observe and investigate their antibacterial properties? Did you observe some difference which spectral range is better for these antimicrobial properties? Depends on the absorbance of these nanoparticles.
Yes, it depends firstly, we use only the tellurium for the first material. So we remove the tellurium for the other nanoparticles because tellurium has the problem that oxidizes very quickly to form tellure oxides. And then they remove completely the emission of the nanoparticles or prevent also the functionalization of the nanoparticles. In the other case, you see the silver of the gold are reduced by a redundant, and this redundant always or sometimes are also the organic molecules that are connected. Okay, the modulation of the spectra comes more from the shape and the size of the nanoparticle, but in this case the shape, than the organic molecule that is detected or that is protected. Because, for example, using an MAP is the same molecule, but as soon as you change the pH, as soon as you change the number of atoms you add, so the seeds that we have for the synthesis, we can modulate the length of the arms of the tips of these nanostars. And then depends of these tips, depends of this size, we modulate the spectra. We can go from the visible up to the near infrared. In this moment, I don't present here the results. One of the last nanoparticles that Javier synthesized can go up to close to 2,500 nanometers. So the solution is almost transparent. You don't see color, but is there. We characterize, and then we see the nanoparticles then inside, but they has not color. And this is good because if you like then to apply, for example, to irradiate in search applications or irradiate in another kind of biological systems, you prevent to burn the biological tissue or to prevent to kill the bacteria by the lasers or everything. But it's not organic molecule. It's more the shape of the nanoparticle.
But what I wants to ask is just the spectral range of these nanoparticles. One is, say, absorbing red light, another in infrared. Which one from them will be better as for antimicrobial inhibiting or killing the bacteria?
Oh, yes. This is depends on the nanoparticles. In one cases, we observe that is better the visible region, but in other cases, is the infrared region. So as much irregular is, you can start it, for example, to the nanorods. A nanorod has two plasmonic bands. Okay, in a nanostarch, we have two. Sometimes we have a small one. You can go to three, but with two always. If you irradiate in the small one, that always is more indivisible. In some cases, the result is very good to have the antibacterial effect. Of course, if you irradiate in the infrared, because sometimes in the intensity is even better, but depends of the nanoparticle. So depends of the molecule we use, the MEP or the the PSS or the polyallylamine, depends of them. But we are very interested in the infrared region or far visible one. So as much as we are over 600 nanometers, better, because it's more stable or more security for the application of for the antibiotics or for the bacteria irradiation or for the cells irradiation.
Thank you very much.
You're welcome.
Thank you. Dear colleagues, more questions? Charles speaker? No. Okay, I have a question concerning stability of your polymeric composites, high concentrated polymeric composites with nanoparticles. Did you observe any migration of nanoparticles from polymer outside? It's very important, for example, for food covering.
Yeah. we don't observe the release of the nanoparticle itself. We observe the release of the ions that the nanoparticle produce with the oxidation. For example, this is one of the effects we like to follow for the intelligent food package, in which we need to control the release because a concentration too much of silver ions could be damage for the food. Okay, but the nanoparticle never release entirely from the polymer. We already have the results with silver and with gold. We are now working with one of the nanomaterials that are better than this, that is copper silver. Copper silver are amazing, the results they are having. The problem is the methodology for synthesis is much more complicated. But I'm convinced that the results in the future will be much better than the pure silver or the pure silver gold. Okay, but we don't see the release of the nanoparticles. We see the release of the products, of oxidation products of the nanoparticles in the case of silver. Okay, but if you use, for example, I show you here the polyestyrene results with the polymer, but we have other results in which we combine 2 or 3 different polymers in order to increase or reduce the network of the polymer to be more or less porosity. And then depends of this mixture of the polymers, you can have one of them that prevent completely the release, and others you increase the release. So depends of this, but not the nanoparticles.
Okay, thank you very much, and thank you for your very impressive.
Thank you.
You can see that we have a lot of questions and very interesting discussion concerning your
Thank you very
materials
much.
and your science. And I would like to say we are waiting for you in Ukraine, especially in Kharkiv, in our institution after the peace in Ukraine.
I hope, and all of you are invited also to visit us here in Portugal, in Caparica. Anatoly and Olesia, that has been here, can tell you that the place is really nice, especially because we have 25 kilometers of beach in front of our city. And this is really very, very amazing, especially in summer. And if any of you like to participate or something, drop me an email. I will share this presentation with Valentin and with Anatoly, so you have then all my contacts. In any case you like to collaborate or participate in the conference or comes here, whatever, so you are welcome.
Thank you very much. So I should go to your country and to the conference because it's a very nice place, I'd like to say.
Yes.
Yeah.
Yes.
Thanks again, and have a nice evening.
Thanks again. Have a nice evening and good luck.
Thank you.
Bye. Thank you.
Bye.